Hello everybody. And so today we have a very interesting topic. I will talk about your income and your genes. Is your income basically written in your DNA? Is it encoded in your DNA? That's a good question, right? We will actually talk about one set of data which was published in Nature paper. And that's this paper. So as you can see, this was, well, it's not a very recent paper. It's but quite a nice paper where these authors we actually claim they first analyzed the genome-wide association studies of actually 300,000 or almost that amount of, almost 300,000 individuals who were chip-seeked, who were actually, genotyped by SNP arrays, and whose genotype data is located in the UK Biobank. So these are people living in the UK, most of them, I guess. And then, of course, many of those people have information about their income, about the household income, as they call it, and that's a quantitative measure, and that's how using simple, quite sophisticated methods of meta-analysis and genetic correlation with traits, the authors revealed a number of SNPs, namely 120 income-associated SNPs. And after this paper, of course, there were a lot of criticism and a lot of opinion. That's a very simple kind of, faced or naive looking at this very complicated matter of income, especially since these authors, if you look at the paper, and especially here, maybe we can again make it a little bit bigger. So as you can see here, what they put here as a sort of line of events, or line of not events, but correlations and causations, I would say. You see this clear arrow from left to right. So they, since these SNPs, they are located in genome of those individuals, and they may change something in genome, and then they claim that it's may change the brain function, which also cause the difference in intelligence, whatever they mean. Well, actually, they mean a particular thing. That's how these people can solve particular tests on, such as IQ tests or something like that. And then this will influence on education, on level of education, which in turn, of course, as they think, will be very directly influencing your income. And that's something, of course, many people basically are considered as an insult sometimes, or even a very wrong assumption. And there was a lot of criticism. But let's have a look. But for us, I mean, of course, all this discussion is very important. But from the practical point of view, what we want to do is now just to look at these SNPs more carefully and look where they are located. And one important picture which we could consider here is this this figure from the paper. Let's make it bigger. So maybe you know what it is. So basically, it's kind of plot of significance of each SNP in terms of p-value. Because that's only one part of significance, towards the influence of that SNP on variability in the income of individuals. And here you see the chromosomes, and each of these line is one SNP. So there's on the chip, there are almost 1 million of SNPs, which were genotyped, and these are chromosome numbers. And you see the highest peaks correspond to the p-value, which is logarithm minus logarithm of 10, which is, if it's 10 minus 16, the p-value like this, top SNPs, they are very significant. But in general, you can cut somewhere here, or maybe even lower. So the authors in that paper, they actually made several cuts. They consider only very significant SNPs. They also considered SNPs which are below -5, p 10 in -5, somewhere here. What I will do today, I will even go a bit lower with this cutoff into 10-4 to also consider some very, little peaks which we can see here. And our focus today will be actually in very low end in chromosome 20. Why is that? Yeah, you will see today, but just to say this famous saying, it's, if something is shining like these peaks, it doesn't mean it's a gold. So maybe real gold is in fact somewhere in the area which is not shining as high areas. So, okay, so from this paper, what we did, we download the data, which is here. So let me open it in Excel table. This was the data from the paper. So here you see actually a big list of SNPs. It's up to 8,000 SNPs. And these are all SNPs which have p-value below 10 and -4. So this is a really big list of SNPs, and we thought, why not consider all of them? And here you see the SNP identifier, which is a RS number. Well, actually, no, that's independent significant SNP. Let me explain it a little bit more. So as you can see, the real SNP, which is was genotyped, was here, and here we have different SNPs. And here, what is not effect allele and effect allele, which means the effect allele. So the position C, for instance, in this SNP means it goes towards increase, or it correlates with increase of income. All right, but independent significant SNP is something different. So those SNPs, as we can see later, they actually sort of clusterized. So they and you actually can see that also on that picture, so that some peaks are really comes along, but most cases they are sort of clusterized. So which means, and that's known effect, that known thing, because because of locus in disequilibrium regions in the chromosomes. So pieces of chromosomes, they are inherited in like full pieces. In the standard European population, we can even see where those chromosome pieces are located approximately, which means out of many of these correlation of p-values which come from individual SNPs are actually associated maybe with one, the biggest SNP, or SNP is the highest p-value, which is considered to be kind of a driver SNP, or independent significant SNP. Still, in our analysis, we considered not just independent SNPs, but we considered all of those SNPs. And the reason of that I will explain later. But so basically what we'll do, we have to upload this table in our software. And actually, as you can see here, the actual data comes from the line 3. And that's we have to remember when we upload the data. Today, we actually will use a special tool for data analysis, which is a pretty automatic, and it's called Genome Enhancer. So that's maybe you've seen it before, maybe not, but I would recommend you to try it. It's very nice and very easy to operate. So I can make the screen like this. And here, what you do, you actually create your project, or you open existing project. For instance, I have here this project related to our coffee break today. And here you can put your data by just drag and drop tables like I presented today. Okay, so actually, we can do that. Let me basically do it right away. So we have this table. Exactly. This is a table, which we just had a look, and we can drag and drop it here. And then you simply click import. And for what it's doing, it's first detecting your format. And of course, what you you have to define what type of omics data you are dealing with. Just a little bit of Genome Enhancer tool. Genome Enhancer tool can actually, Genome Enhancer, we call it without even tool, it can manage working with many different omics data. So of course, a very standard is transcriptomics data, or proteomics data, or ChIP-seq data. We had about that many discussions before. But you can also upload genomic data, and GWAS data considered as genomic. Basically, they define which are changes in genome or which variations in genome we are analyzing. And so you simply then have to define what kind of omics we are working with. It's a genomics data, right? And okay, let's put another name of the table, because I did upload it already, the table before. And here we have to, well, it's already recognized the header is on line 2, and the first row is in line 3. But actually, what we have to do is we would select here, generate unique ID. And the reason is, well, actually, we shouldn't do this. We can directly select the column which we want to use as an identifier. So here we use targeted SNP as identifier. So it recognizes this is a type of data is actually SNPs. And then we import this table into our system. Actually, I will switch now to the platform view of that tool, because before we actually start, you can start, in fact, right away by just clicking next, 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 and that's it. So, and it will run this tool under certain default assumptions. But before we're doing that, let me show you the data itself, and we'll think a little bit about what kind of assumptions we would like to consider. But before going into that, so first, let me go next, and just to show you this this table, which we just uploaded. So this was the table. So you see what we have here is, again, the same Excel table, but and all the columns are here. And especially what is important is this p-value, which is defined not for all of the SNPs, because some of the SNPs actually are not considered, well, the p-value were not calculated in this table. So we got fewer number of SNPs with p-values calculated. And you see these like SNPs with a very low p-value, which is very significant, or like ones which in 10⁻⁴, so those which less significant. But we want to consider them all. And the reason is, let me actually show you some of the slides I have prepared, which explain a little bit what we want to do. So the usual consideration, what people are using to analyze such SNP data is, first of all, okay, these are SNPs, and as you can see and you can expect, majority, basically all of them, 100% of those SNPs which are found to be here as significantly associated with house income, are located not in the coding regions of gene. So it would be very rare if you have like a mutation of a gene which changes something in the protein, and you have a mutated protein. So this usually causes very big changes in genome and in physiology. So those slight changes here we consider as normal, healthy people, individuals, and most of the SNPs, and majority, in this case, 100% of the SNPs, are located somewhere outside of the genes. I mean, outside of the coding regions of genes, right? So they are often located in regulated region. And it's not very clear, and very little is known what is happening if letter A, for instance, here in some region in genome, which doesn't encode any gene, any protein, changes to C. What happens? And often people doesn't even actually consider these consequences. They just say, okay, there is a SNP near a certain gene, and that let's consider that gene as important. And what this SNP is doing to that gene is not really considered or discussed. Here we want to look at that very specifically because what happens, little changes in nucleotide, just one little letter A to letter C, can actually delete or kill a particular transcription factor binding site. Like in this case, and that's known, SNPs actually even related to very severe diseases like diabetes. Such SNP which is also correlated or linked to this disease. In this place, just change A to C. In those person, they lose a site for a particular transcription factor. So the consensus of that site is fitting very well to the sequence with a letter A, but when it's changed to C, it's not fitting any longer. Or one SNP can actually lead to a gain of a site. So there was no site here, but after the change, you get the site. Or the same, one change can actually in the same time delete the site and create a new site. So like in this case, again, for diabetes, and here one change deleted, removed a site for EGR1 and CP2, and in the same time created a CABP site in the same place. So in such a play with transcription factor binding sites, well, although maybe very little, but can influence on how transcription factors are actually binding to this place. And sometimes the effect can be quite severe, like in this case, you see very severe diseases. And actually, to explain why we have to consider all the SNPs rather than just one most important SNP is somehow explained on this picture, where as I consider, if you have a transcription factor binding to its binding site to a genome, if you have a nucleotide change in the transcription factor, so basically you change the protein, and it will influence hundreds of thousands of genes, because this transcription factor is regulating many genes, especially such factor like AP1, which is very ubiquitous. But if you have a change just in one nucleotide in the binding site of the gene, it will influence just maybe some regions, some genes nearby. And also we know that actually the regulation of genes is very flexible. So we see very often when a binding site disappears, this same transcription factor can bind to a site nearby. So which means it's quite flexible in binding and still be able to regulate the genes. So, and we often see if particular gene is regulated by, for instance, AP1, then we often see many transcription factor binding sites for AP1 in one region, which creates some sort of redundancy. So if one site or another site is removed through some genetic or epigenetic changes, still factor can bind to that place and regulate the gene. And that's how I illustrate here. So like if you consider DNA as a road, and if you consider a transcription factor as a kind of a car, if you have a brake in a car engine, so nowhere you go nowhere, right? But if you have one kind of arrow on the road, you still can drive by just avoiding it. And the only way when everything is stuck, when the whole road is blocked. So when you have like a massive changes in many places in the area, and then you got stuck. So actually, this analogy tells you that that's why we consider not just one SNP, but all the SNPs which can happen in the area, because many of them can influence on DNA binding sites. And if they influence in the same direction, then we can claim that maybe the real effect on the nearby gene is pretty high. Just to remind you what we are talking about when we talk about transcription factor binding sites. So, you know, in TRANSFAC, we have those positional weight matrices, and it's not the only database, but in TRANSFAC, we kind of try to collect all information, or all available information in the literature and in the other databases. So it's very comprehensive collection of PVMs. And they are built by collecting known sites for a particular factor. And then these PVMs are used to predict where the potentially transcription factor can bind. For instance, we can predict them in the promoter of that gene or in enhancer of that gene, and we'll talk about this a bit later. But then if something happens like a SNP in this area, and for instance, here there was a nucleotide G, but it changes to T. And since this nucleotide is very conserved, which means it's always has to be G in this sites, but it changes to T, so they score when you calculate it for one variant of SNP and for another variant of SNP, the difference between scores can be very high. So this delta. So here we get a score which is higher than threshold, and here we can get a score which is lower than threshold, and then basically we interpret it as a loss of that site. So we can count a number of such events in one area or in whole genome, and we can say, okay, we see a systematic change. For instance, many of those SNPs which are related, for instance, to a certain trait, they are changing very significantly, for instance, sites for NF-kappa B. And we'll see those statistics in the output. All right, so another approach which is complementing this approach of considering one particular, what changes actually happens when exactly at the place of the SNP, so when SNP is touching the transcription factor binding site itself. Here we consider the whole area, not a big area, but area around the SNPs. Because as we can see, those SNPs are indeed located in the clusters, but also we know that transcription factors are working together. So if this factor cannot bind any longer to this place, then it cannot interact to another factor, and which would have to be interacting. And well, if that happens, then the whole regulation doesn't work. So we have to consider these areas from a point of view of site combinations, and from a point of view of which kind of complex formation of transcription factor complexes, so-called enantiosomes, those SNPs can influence. And that's done with various algorithms which are in our platform. One is just counting number of sites found in the areas around the SNPs and compared it to the other SNPs. So basically we take them as a control set, other SNPs in genome, which are not related to my particular trait, in this case to increase of the level of the level of your income. And then we count how many times we observe a particular site in areas of our interest, in areas around the SNPs which are correlated with our trait, and compare them with areas around other SNPs. And with the simple statistics, we can estimate which of the sites are actually considered to be enriched. There is even more complicated story, and I will not go into details in that, but let me. Okay, no, it's not here. But okay, so again, last slide here for today, before we go directly into the result, is this thing. So as you can see, what we are going to do today is we will look at those SNPs. We'll check which of them is actually changing or losing, leads to change or gain or loss of particular transcription factor binding sites. And then we look at the areas around those SNPs, considering which transcription factor binding sites are enriched in these areas and in which combinations. And then from that, we will come up to a list of transcription factors, we believe, whose sites are affected in the genes, which might be regulated by these areas. So we have to consider also genes which might be regulated, and we'll look at that next. And then when we have those transcription factors, we can go into network analysis, and that's what system is doing. And in order to find so-called master regulators, so such molecules in the cells, like receptors or ligands or hormones, as we'll see today, which may play a role as a master regulator, which means they can influence activity of many factors, which altogether are considered the binding factors around those SNPs. And to do this analysis, we actually need two types of data. We have these SNPs, their positions. That's we have for sure. You've seen this table, but we need also actually a gene list. We need those genes, we believe, which are expression is considered, is regulated by those SNPs. And that's the most tricky part. Let's have a look then into the data, exactly into the data in our tool. So as you can see here, you see a little bit of different interface. So we were looking at this, in Genome Enhancer, but now we want to change it to a different interface, which is called the platform interface. So this is basically Genome Enhancer is a big pipeline, which is doing what I just explained. So it's looking for binding sites around the SNPs in this case, and then finding transcription factors, and then build this master regulator cascade. But beyond, on the basis of that tool, we have a so-called platform, which collect all the tools which are used in that Genome Enhancer. And in this platform, we can look exactly into the data and do whatever we like with this data. Again, we can open this table, which we just have seen before. And this is a table of SNPs. That's fine. But actually, what we want to convert it, we want to convert it into a table, basically into the positions in genome. We have those positions which are here, and those positions can be also calculated automatically from the SNP number. But actually, those positions are given in this table in the previous genome build. So it's this paper is from 2019, and they were doing still in the build HG19. And what we want to do now, we want to actually do it in the build 38. So you have to convert those positions into build 38, and that's what we are going to do using our tools here. We have, you see, methods which do various types of conversion table to from one build to another. So we have these very detailed steps. Let me just show what we get at the end. So actually, after such a conversion, so we can have a look how we did this conversion. So basically, if you see such a file here, which have a little icon as an arrow, which means this is already is converted to a position into genome. You can even see it by double clicking on it. So it actually opens a genome browser, namely it opens exactly the genome browser in that particular genome build is HG19. And you can put also a genome on that genome browser. Yeah, you can have a look which ones. All right, so that's the previous build. So we have here, we can put here genes, for instance, and then we can see where those SNPs are located. These SNPs are, for instance, located in the end of that gene, and so on. So we can consider where they're located. But still, this is a HG19, and we want to convert those SNPs into HG38, and that's done with a conversion tool, which is located here in our tool box among the galaxy. We have these liftover tools. That's a very standard thing. So like you can lift over just the simple coordinates, or you can lift over VCF, which means the data like this. So the SNPs are the sort of VCF type of data. And by converting liftover VCF, we actually converted all this into this list of SNPs. And you see that's already opening in another build of the chromosome. And in total, we can see this is 5,880 SNPs, which we now consider for our analysis. And then we mark this as a genome, as a genomic data, because with such a mark, now this data can be imported into genome browser, and then you can start analysis of those SNPs in genome browser. So how to do that? You have such a file. You right mouse click, and here you can set the genomic type as a genomics, and so that's how we got this genomic type of data. All right. So, but that's only one part of the work. So, as I said, to start the Genome Enhancer, we need also a list of genes. And the easiest way of converting it to these SNPs into genes is actually simply to consider those genes which are located nearby. But the problem is, what is nearby? So to do that, we have this track to gene set function, which is located here in among the methods. And what you do, you simply drag and drop your SNPs here, and you select, what kind of output you want. You want to see which SNPs are located introns, exons, in 5' or 3' areas of the genes. And then you give a file name for the gene output, but then you have to consider what should be the area which you consider that SNPs can influence expression of that gene. And by default, we usually take very small, and not only us, people are using also in that paper, they're using very narrow regions. So they consider only those SNPs which are located in the promoter area of that gene, in intron of that gene, or very near from the 3' end, like not more than 1,000 kb, or maybe up to 10,000 kb. That's considered the most important regulatory regions for the genes. And that's true. Although you know that genes can be regulated by very distant enhancers. And by removing those SNPs which are located very far, quite far away from genes, but still can be influencing expression of these genes from the long distance connection, we actually lose a lot of information. That's why what I did here, I considered left and right a very big area, up to 300K. So 300,000 nucleotide upstream and downstream from the gene, including the gene itself. Because that's approximate areas where we can consider might be enhancers for that gene. And if you see SNPs located in those enhancers, they may be influencing my genes. And of course, such a list of genes gets quite big. So we got actually 790 genes. And here, by the way, you get this nice picture where those SNPs are located. So the blue bars are the representations of those genes. So the blue bars are exons and arcs are introns. And this little part here left is actually 300 kb upstream area, and here 300 kb downstream area of the gene. And these right bars are SNPs, and very often you see them in cluster, like here. So they are located nearby. And often they located in 5' or 3' areas. Sometimes they located inside the gene. And here you see the counts, how many SNPs, and where they exactly located. So, but this gene list is quite big, and maybe it's too big to. It's like the hypothesis about that those SNPs are influencing all these genes is very vague, because it's just a distance. So what people in this paper did, they actually also considered so-called EQTLs. So there is a database reporting about a connection between particular SNP and expression of that gene. But these experiments, they always done in different tissues, different cell lines, and those EQTL connections are very particular. So they are working in a particular tissue or cell type. Here, in case of income, it's very hard to consider what kind of tissue or organ is most important for my income. But consider this, but let's first before doing any assumption, let's do the following. What we can do, we can take all these genes, these 790 genes, and run them against tissues. What I mean is just the following. We have here a folder where I actually created a folder, and that's a very flexible format which you can do as well, where you put just lists of genes. Here there are 61 lists of genes which are corresponding to of genes expressed in particular tissue. We just took this information from Protein Atlas. So for instance, these are 14,000 genes which are expressed in adipose tissue, or for example, other set of genes, which is another 14,000, which is expressed in colon. So these are 61 general tissues or tissue parts of our body. And then what you can do is actually you run comparison. You have your gene list, like these 790 genes, and then you can map them to this list of genes. Whatever list you have, you can prepare it yourself. And then the system will calculate the enrichment. So how to do that? Just let me demonstrate it. Very easy. So we have here the functional classification tool. You click here and you put your gene list. This is gene list. You put it here. And then you can run it against gene ontology, other ontologies, and this you may know. But here we have this special function called repository folder. And now you can put here any folder of your, as you like, where you have collections of genes you want to compare to. One important actually thing which you have to also put is a reference collection, which means overall collection of genes we are considering. And here we have to put the collection of genes which are have at least some data about gene expression in particular tissues. So basically those genes which are from Protein Atlas, we have this table of all gene expression, which is 19,000 genes. So basically all genes where the gene expression value were measured in different tissues. So we have to put this table as a reference table so to calculate these p-values. And then it's have a p-value cutoff, and you simply run it. So it runs very quickly, and then you get such list of such output. And what you have here, the p-value, an adjusted p-value. This is a p-value of enrichment of a particular tissue expression in your gene list. So we have around 790 genes, and you see what we have here: cerebellum, spinal cord, cerebral cortex, testis, endometrium, basal ganglia, so we see hypothalamus, well, among others, but I must say the top of those guys who have enrichment are different parts of the brain. And that's also the same assumption. This same conclusion was done in that paper. They did a similar thing, although with a different set of genes. So they considered a smaller set of genes because they look at only very highly enriched, highly important, and with a p-value, very significant SNPs only. Here we have a bigger list of SNPs and a bigger list of genes, but still we see this tendency that especially cerebellum is very much enriched here. So, okay, so we see that among those genes which are near, well, where the clusters of SNPs can influence expression of the genes, many of them are expressed in cerebellum, in brain in general. So that's how we did this. The next, so actually we can annotate those genes by expression from various parts of brain, like cerebellum, hypothalamus, midbrain, and other. And then we filter only those genes which have at least some expression. Some means at least higher than 1 unit from Protein Atlas values. And we got here 300 genes which have among those genes which are located near our SNPs and expressed in brain. So that's a set of genes we want to work with. So basically, the idea is look at which SNPs from what we have now in our hands actually can influence on expression of genes nearby and which we know expressed in brain. All right, so these are two sets of genes, and that's been now returning back to Genome Enhancer like we did before. And so in Genome Enhancer, you can look other maybe recordings, what are the steps to run Genome Enhancers. So basically you have to put here your data like I did. So this SNP data, the table data, then you put next, and then you create sort of a starting collection. And the collection I was working further is this one, where I put this SNPs, you see they're red here, and I put this list of genes which are these 300 genes which are located near those SNPs and expressed in brain. And then I click next. And here, you basically what I need, I selected this collection, this condition we call it. I considered just this one because in general, in Genome Enhancer, you can consider many different conditions. And then I have to select here disease. Okay, what kind of disease is correlated with your income? It's, well, we don't know. That's why, so basically, just from retrospectively and from that paper, we also see that those SNPs which are influencing your income can also be connected to very different diseases. Mental diseases, but not many, few. So that's why what we can put here, some mental disorders. Like mental disorders, for instance, or some other disorders of the brain. So this type of input is just to see something at the end of our analysis, which may be related to what we are talking about, how those SNPs are also, connected to disease areas. And then we start analysis, and that this analysis will take a couple of hours. It will do everything what I told before. So looking at transcription factor binding sites, which are changing and which are enriched, and also looking at the upstream and finding master regulators. So what we will do now, we will actually go to the end, and we'll open the report which is generated by the Genome Enhancer, and we'll look at it in the rest of our time. All right, so as you can see, this report considered the most important transcription factors which are found enriched or changed due to those SNPs. And it's actually also look at these master regulators, most important, most interesting master regulators, which are which were found. Okay, so but let's go first step by step. See, this is the same picture which I showed before. This is a table of those genes. And first what it did, it actually took those genes and put them, start comparing them to different ontologies, for instance, to gene ontology. And you see here some interesting thing. For instance, you see reproductive behavior, how your income connected to reproductive behavior. So that's something we have to think about, but let's have a look further. So we have different intracellular pathways which are connected to those genes, like including also EGF pathway. So that's something which also discussed in the paper, which we're under analysis. And then to which diseases those genes are connected to as a biomarkers. Many are connected to very strange diseases like lung diseases, artery diseases. We don't see here. Well, we see here prion disease. So some of them connected to that, but we can actually have a look more in detail about those diseases maybe later. But now what we want to see is how those SNPs are affecting my sites. And here you see the p-values of the gain and loss, as I explained to you. And if you look here, for instance, such factors like rel A, this is the NF-kappa B factor, and it's very significantly enriched in terms of gain. So many of those SNPs actually lead to creation of rel A site. In the case when the nucleotide, you know, there's a effect nucleotide, non-effect nucleotide, and effect nucleotide. So those nucleotides which are connected to the effect, which means which are connected to higher income, they are quite often and significantly located in creating rel A transcription factor binding sites, or well, for instance, E2F4. So what does it mean? So okay, some sites are changing, some factors may be more often binding to those places. So what? So then next, what we started to do is, as I said, we start looking at these areas nearby. So we may see it later. So, but this is something what the output is showing. So the areas nearby those SNPs enriched by these transcription factors altogether. So we can consider them as a clusters of factors, so enhanciosomes, which are affected by those SNPs. And this histogram shows how the areas around the SNPs correlated with income are enriched by these transcription factor binding sites compared to the areas around other SNPs, which are not correlated with the income. All right, so but at the end, what I want to really look at this master regulator picture, let's open it in a bigger window. So what do we see here? We see here in pink are the master regulators. So and what do I mean? So but let's start from the bottom. So here are the genes which are potentially affected by our SNPs, right? So we don't know whether they're really affected, but they are located nearby. And we assume they are affected. Then here are transcription factors, which we found are clusterized in the areas around those SNPs. And here above those transcription factors, we see this cascade of events, which ends up at the top by master regulators. So molecules in the cell which can influence activity of those factors. And what is important, they are actually expressed by those genes. You see these dotted lines, so-called feedback loops, right? So all of those master regulators are expressed by those genes itself, which means this can be auto-proliferative loops. So that's the whole story about these feedback loops. But so this is one of the important part, how we actually consider those master regulators. And our focus, you see many master regulators here, not many, but few. But our focus was on this little protein, which is called oxytocin. So that's a famous love hormone. And it's appeared to be, here it found to be as a master regulator. Of course, not the only one. But if you look even further, by when the system started to consider those master regulators more in a hierarchical way to each other, actually it even put this oxytocin on the top of everything, because topologically it can be buff of all those master regulators which we found here. So that's appeared to be an important master regulator. And that was very interesting and intriguing for us. In YouTube, I recorded one small film, which actually reports about a study of one American neuroeconomist called Paul Zak. And he actually claimed that oxytocin, not only him, but it was also a very nice experiment he did. He claims that it's not only a love hormone, but it's actually a trust hormone. So what it means, he is a neuroeconomist, so he makes various games with people. So he had the game where he had two players, like one person has to give money to another person. So there were more complex rules, but basically at the end he has to give some money, which is well given to this person. And then what he found out that actually there were people who giving more money and people who were actually avoiding to give money to other people. And he compared those, he took blood, just some sample of blood from those players and tested this blood. And in terms of the level of oxytocin in the blood. And he found a very clear correlation between level of oxytocin in the blood of the player and the amount of money or proportion of the money this person is able to give to another person. And that's why he called it trust. So those people who have more trust to other people, they seem to have higher level of oxytocin in their blood. Moreover, he actually made the following. He gave to those people before the play to some, he divided the set of players into test set and the placebo set, and then the test set, what they did, they have like an inhalator with oxytocin, and they were inhaling it before they play. And then Paul actually compared the result, and he found that actually those people who spray this oxytocin, they increased the amount of money they give significantly, well, it was significant level on 17%, maybe not big, but it was significant. So which means really it's a trust hormone. And here what we see, as we show in our results, that it's a master regulator of those SNPs which are correlated with your income. So does it make sense? I guess so. So there is some connection between trust of people, people who have trust to other people, and ability of those people to get maybe richer or get more income, or maybe get more convincing to get more income. So that's something which is very worth to consider. And that's what I want to talk to you today. And I guess I will finish with that. Of course, the main idea of the presentation today to show you how we can analyze SNPs, and basically it doesn't matter whether the SNPs are coming from GWAS studies of a particular disease or of such trait which is not related to anything in terms of disease, it seems, but actually it's can be also correlated with your genome and with SNPs in your genome. As you can see, there is some chances that genome is your income is encoded in your genome. So I have a question. GWAS generally tests about collaborating SNPs resulting in particular trait. Can TFs form kind of basis of such an SNP trait interplay? Can the geneXplain predict which TFs participate? All right. Yeah, you said this word, collaborating. And that's exactly what we are doing here. Although it's maybe on a small scale, but not really. So maybe I can demonstrate it on one slide I have prepared for you. So I think that's here. So as you remember, one part of our analysis is looking for these composite modules. So basically, that's exactly what you're saying. Collaborating transcription factors can collaborate. But you were talking about collaborating different SNPs to each other. But so far, what we are talking about, if we have one SNP, for instance, like in this case, one SNP located in some regulatory area, and it affects collaboration or cooperation of transcription factors. So the same transcription factors are acting in different places, right? In this place, they are collaborating in this way, cooperating in this way, in another place, in this way. Here we got the different different position of a SNP, so we don't have here binding of those factors altogether because maybe one important binding site is destroyed, and this whole complex cannot be formed. So in that sense, through this same set of factors, like through this hierarchy, you can observe cooperation between different SNPs, what you are talking about. So so which means it's also shown in this picture. So basically, these different genes, which are located near different SNPs, they all together may play a particular role in one pathway. And that's what we were building when we were building master regulator pathways. So we were putting all those genes together, which means genes which have those SNPs nearby. So, and these genes are collaborating to each other in one pathway, which means maybe one person has this SNP in a state of of effect. Another person has this SNP in effect of effect, in the state of effect. But altogether, they're playing different roles in one pathway. And if this pathway is signaling pathway, it's often the case that any component of signaling pathway is actually, in fact, affecting the result. It's maybe different a little bit towards metabolic pathways, where it's important to consider rate limiting steps. So there are genes in one metabolic pathway, so proteins, enzymes, which may have different level of expression, but still not influencing on the output of the pathway, because there is one maybe rate limiting enzyme and SNPs in that gene. And only those SNPs are important for that outcome. In case of signaling pathways, it's because it's more like a hierarchical and parallel. It's very often any of those proteins which are a part of those pathway have a kind of cumulative effect on the final result of functioning of that pathway. So basically, SNPs are collaborating in pathways, and that's what our tool is doing. First, it finds those transcription factors which are cooperating on those SNPs, and then it finds those pathways which are connecting those transcription factors all together under the roof of a particular master regulators. So that's why we are talking about a SNP collaboration. Okay, I have another question. SNPs occurring in the overexpressed genes only taken into consideration. It's a question like SNPs occurring in the overexpressed genes only taken into consideration, or the percentage of the SNPs in the underexpressed genes also hold importance when we look at the SNP and genes, like the brain tissues in the case of discussed here. Very good question. Thank you very much. Of course, the best setup would be to have a SNP data like from GWAS, which are connecting SNPs to a particular trait. And if we will have a trait which is very specific to one tissue, I don't know, some muscle dystrophy or something related to one tissue, we may have expression data from genes in that particular tissue, right? And then we can talk about upregulation and downregulation. Well, again, downregulation compared to what? So to the norm? Yeah, maybe we can consider that. But often such complex traits like income or like educational attainment. I had another recorded seminar about educational attainment. This very complex trait, and we don't know in which tissues. Well, we can make an assumption in brain, but which part of brain? And that's it's a very big assumption. Not only brain, actually many different. If my hands doesn't work, so I cannot do anything, and probably I will not have a big income. Who knows? So we don't know in which tissue we have to consider high or low. But okay, if you look at in the brain, like in this case, what I did, I just take all genes which are at least expressed. But of course, we can see genes which are highly expressed and genes which are expressed low. And we can, in fact, compare to other tissues and consider maybe low expressed genes as well. And see and look at those SNPs and maybe compare those SNPs which are connected to the genes which are highly expressed and those which are low expressed. We don't know. In fact, in such a case, when we just have SNPs, we don't know whether they're influencing on upregulation or actually influencing on repression. And we know many cases when the same factor can be activated for one gene, but repressor for another gene. There are now very few universal repressors or universal activators in terms of transcription factors. So that's why it's very complex to consider it without actual expression data. But in principle, your question is right. So once we have expression data and very focused tissue or maybe cell type to look at, and maybe trait which is considered, which is related to some kind of changes. For instance, we have a disease and we can compare expression in disease versus normal, right? Then in that case, we have upregulated genes and downregulated genes. If you have just the brain of normal people, then of course we don't know which gene is upregulated or downregulated compared to what. Yeah, we need to have a comparison to know which is up, which is down. But generally, this is very worth to consider. SNPs may influence also, as we can see, repression of the genes. Okay, any further questions? Okay, in the meantime, I just a little bit interesting thing. As I mentioned, I have recorded one seminar about SNPs found to be correlated with educational attainment. So, which is basically the higher level of education or level of degree which person is having. So, and then it's also quite such a social thing, but there are also other types of SNPs which are connected to those to this trait. And interesting that as in this paper which we were discussing today, they found a correlation between SNPs which are connected to educational attainment and level of income. And in my study, in my analysis, I did compare them. I cannot say so. They are quite distinct. There are few SNPs which are the same, but very few. And in terms of genes, well, actually, I can show also that. Here in the platform, I did analysis of those connections. So, for instance, if we just compare genes which are connected to income and connected to education, so these are educational genes, and here income genes. Just from the number of genes, actually, very interesting consideration. We have quite much more genes connected to education attainment rather than connected to income. And that was very striking. Actually, in terms of number of SNPs, we can actually have much lower number of SNPs in case of educational attainment. But they are all kind of distributed among genome and touching many genes. In case of income, we have a lot of SNPs, quite a number of SNPs, but they're quite clustered and they're affecting very much lower number of genes. Maybe you can consider that income is sort of only one type of way of having your education. Education can be in many different fields, but maybe only very focused type of education is really related to your income. And we know quite well some type of education maybe not really not really connected to a good income, and some type of education really very much connected to income, like education leading you to work in a bank, for instance. I don't know. Anyway, so this is interesting consideration, but in terms of common number of genes, there are few. I also compared which common diseases and found none. Yeah. So actually, I don't think so that there is a much correlation between these two types of SNPs, but still there is some, yeah, as it is described in the paper. And if you don't have any further question, let me finish for today. Thank you very much and goodbye.